Hello everyone, welcome to the Brewing Science and Technology series. I'm Amul Gimire. Today we shall discuss about the massing theory and the biochemistry behind the massing. Massing is a stage in the wort production in which the grist is mixed with the brew water and the resulting mass is held for a predetermined period at a defined controlled temperature and controlled pH. Massing is very crucial because it impacts its performance, filtration, beer flavor, alcohol content, and foam stability. Why do we need to mass? So massing has two purposes. One is to dissolve as much of insoluble compounds to the soluble compounds and get more yield from the raw material that we use. This is performed by continued degradation process that were started during malting, which includes breakdown of starch, protein, and beta glucans. The second objective of mass is to get better quality of the extract. Some of the desirable components such as sugar and protein are extracted more, whereas the undesirable components such as tannins in the husk are extracted less. What are the goals during massing? We should have optimum mixing of the malt, grist, adjunct, and water for homogeneous mixture. The agitator speed of the mass tun needs to be optimized so that the solution is homogenized. However, care must be taken to avoid the ingress of oxygen due to the vortex effect formed at high agitator speed. We should have maximum enzymatic reaction for the conversion of complex molecule to the simpler dissolved form, which shall ultimately result in higher yield. The mass is held at right temperature and pH for sufficient time so that the enzymatic reactions are completed. Massing affects various downstream processes. Massing impacts the spectrum of the sugar present in the wort, which has final effect on yeast performance, alcohol content, and flavor profile. Higher maltose will result in higher attenuation of the wort, resulting in higher alcohol production during fermentation. Similarly, the spectrum of peptides affect the foam stability and the colloidal stability. Massing at higher agitation condition may solubilize the beta-glucan from the grist and may consequently increase the viscosity and the runoff time during the wort filtration in the lauter tun or mass filter. Therefore, massing is optimized in order to have positive influence on the downstream processes. The final wort composition depends on the time temperature profile. The goal when choosing a suitable time temperature profile is to produce a wort with the desired properties and flavor. Massing involves heating at various temperatures for varying time and is very energy intensive. Different approaches such as use of hot water and other renewable energy are explored for heating in order to be energy sufficient. So in a bigger picture, massing is mixing of water and malt to produce spent grain and wort. During massing, the malt grist, solid adjuncts, and water are mixed at suitable temperature and time for the malt enzyme to convert various cereal components into fermentable sugar and other nutrients. Spent grain constitute the solid residue that remains after wort has been separated from the mass. Spent grain contains a high proportion of polysaccharide cell wall material from the malt and adjunct with some lipid and proteins. In addition, a proportion of entrained wort always remains in the spent grain. The liquid separated from the spent grain containing the sugar and the yeast nutrients is referred to as wort or extract. So what is extract? Extract is a measure of wort concentration. Extract includes the substance dissolved in water from the brewing material, such as malt and adjunct, by the massing process with a suitable time temperature profile through the action of enzymes. Extract contains the substrate for the yeast, such as fermentable sugars, free amino nitrogens, minerals, vitamins, and other yeast nutrients. And other than the substrate for the yeast, it also contains other wort constituents to produce desired beer, which includes unfermentable sugar and dextrins, soluble protein, inorganic substances, and flavor and color compounds. Each style of beer demands its own wort composition, and thus the massing process needs to be modified to achieve so. So where does all that grain go? Here we see where does all that grain that we use for massing go. Major portion, almost 80% of the grain, is soluble as extract that constitute the fermentable and the unfermentable extract. Almost 40 to 45% of the grain is fermentable extract, which is utilized by the yeast, and almost 25% of the grain is unfermentable extract, which gives body and mouthfeel to the finished beer. Nearly 20% of the grain is separated from the wort as a spent grain, which includes the husk and the insoluble protein. Some extract is always entrapped in the spent grain, and some are lost during the transfer and pipeline push out to different vessels. In total, the extract loss accounts for 5 to 10% of the total grain solids. We see here different units for the measurement of the extract. Boiling, bricks, and plateau are three different units that are used interchangeably, but all of them refer to the same thing. They all express weight percentage of sucrose solution. Here sucrose, mean table sugar or the common sugar that we use. Boiling was published in 1843 as a unit of concentration for Wort and sugar solution based on the density measured at 17.5 degrees centigrade, whereas degree bricks is also used as a measure of dissolved solid in weight basis, but it was originally measured at 15.5 degrees centigrade. However, these days it is measured at 20 degrees centigrade. It was based on a recalculation of the table of boiling. The scale is used principally for expressing the concentration of sugar solution, as in the case of syrups commonly used in brewing. It is widely used in fruit juice concentration and winery, but limitedly used in brewing. Plateau scale is a revision of that of boiling and made by Plateau in 1918, since the degree boiling was erroneous. As with the boiling table, the concentration usually of wort is compared to the value of sucrose solution measured at 20 degrees centigrade. So the difference here is boiling is measured at 17.5 degrees centigrade, whereas Plateau is measured at 20 degrees centigrade or 68 degrees Fahrenheit. The concentration in degree Plateau is numerically roughly equivalent to quarter of the present gravity as depicted by the equation here. So if you see, the specific gravity can be calculated by the formula 1 plus Plateau into 4 by 1000. For example, for 12 degree Plateau, the specific gravity would be calculated as 1.048. So the equation can be fairly used as an estimate between specific gravity and Plateau. Understanding the extract is very crucial in brewing profession. Extract, like we saw before, is measure of wort concentration. The concentration of all soluble constituent in wort before the fermentation starts is called original extract, for example here. However, once the fermentation starts, the presence of yeast and the resultant formation of ethanol have effect on extract measurement that require correction. Since ethanol is less dense than water, it exerts a depressing effect, and for this reason, uncorrected values lower than the actual extract are referred as apparent extract. Concentration of the extract measured predictably after the removal of ethanol by distillation and correction for volume and temperature are referred as real extract, which is this value here. So if you see, apparent extract is somewhere in the range of 2.5 to 3, the real extract is in the range of around 6. So real extract is always higher than the apparent extract. Apparent extract is lower because of the depressing effect of ethanol formed during the fermentation process. This is the relation between the original extract, real extract, and the alcohol percentage on weight by weight basis. This equation has also been defined by Boiling in 1843 and can be used as a fair estimate to calculate the relation between original extract, real extract, and alcohol percentage. In this equation, P here is the original extract, ER is the real extract, and A is the alcohol content on weight by weight basis. Extract composition of wort. We shall see now the biochemistry of massing at a molecular level. Carbohydrate accounts for 90 to 92% of the total solid in the wort. Maltose, 2 units of glucose polymer, is the most abundant sugar in the wort, which accounts for almost 40% of the total solids. Maltotriose and maltotetrose, which are respectively 3 glucose and 4 glucose carbohydrate, form about 20% of the total solid. Simple fermentable carbohydrates such as glucose, fructose, and sucrose composite about 15% of the total solid, while the longer chain dextrins, glucans, and pentosanes, which is a C5 sugar, composite around 20% of the total solid. The same is also seen here in this graph. If you see maltose, it composes the most of the extract, whereas the glucose, sucrose, fructose, maltose, and maltodextrins form smaller proportions. The non-fermentable extract, which include dextrin and other insoluble proteins, composites about 20% of the total solids. Carbohydrate are biological molecules consisting of carbon, hydrogen, and oxygen atoms. Depending on the number of the carbon atoms, carbohydrates are classified as monosaccharide, 1 sugar molecule, disaccharide, 2 sugar molecule, oligosaccharide, 3 to 9 sugar molecule, and polysaccharides greater than 9 sugar molecule. Monosaccharides are simplest unit with the general formula CS2ON, where N is between 3 and 6. If N is 3, it's triose. If N is 4, it's tetrose. If N is 5, it's pentose. And if N is 6, it's hexose. For N is equal to 6, hexosugars such as glucose and fructose molecular formula would be C6H12O6. Easy way to differentiate between fructose and glucose is that glucose makes a 6-carbon ring, 6-membered ring, whereas fructose makes a 5-membered ring. You can see here 1, 2, 3, 4, 5-membered ring for fructose, and for glucose it's 1, 2, 3, 4, 5, 6-membered ring. Glucose are called alpha and beta based on the arrangement of the hydroxyl group. So if you see here, these hydroxyl group here, these and these, if both hydroxyl group are on the same side, it's called alpha. For example, in this case, alpha glucose. Whereas if this hydroxyl group is present on the other side, and since this hydroxyl group and this hydroxyl group are on the different side, it would be called beta glucose. Disaccharides are two monosaccharides linked together by glycosidic bond. Example of disaccharides are sucrose and maltose. Sucrose consists of 1 glucose and 1 fructose unit. For example, here, the 6-carbon ring glucose and 5-carbon ring fructose, they are bonded together by a glycosidic bond, and this is called sucrose. It's found in a small quantity and can be added as an adjunct to the ketone. Maltose is 2 glucose units. For example, here, the glucose and glucose, most important sugar in the wort, that makes up around 60% of the wort sugar. Maltose is found in germinating seed, example barley, as the breakdown there is starch stored to use for food. Trisaccharides and dextrins. Similarly, maltotriose are oligosaccharides with 3 glucose units, and they are partially utilized by the brewing yeast. These are 3 glucose units and therefore called trisaccharides. Dextrin represents almost 90% of the non-fermentable residue in beer. Some 40 to 50% of the wort dextrin contain 4 to 9 glucose units, while the remaining higher dextrin contain 10 or more glucose units. Dextrin are glucose polymer and can be linear with alpha-1,4 linkage or branched with alpha-1,6 linkage. For example, here, this is numbered as 1 carbon, 1, 2, 3, 4, 5, and 6. This is always numbered in this way in glucose. 1, 2, 3, 4, 5, and 6. So if you see the bond here, the glycosidic bond, it is formed between 1 and 4 and therefore called 1,4 glycosidic bond. Similarly, if this bond is formed between 1 and 6 carbon atom, this is 1 and this is 6 carbon atom, this bond would be called 1,6 glycosidic bond. So 1,4 or 1,6 are bonds which depicts the number of carbon atom involved in bonding. Dextrins are not fermented by the brewing yeast, and they persist in beer where they contribute mouthfeel and body. Concentration of dextrin in wort ranges from 2.5 to 4.5 gram per 100 ml of wort. Starch. Starch is major carbon storage material of grain plants. Chemically, it is a polymer of glucose in which maltose is the repeating unit. The sources of starch are cereals that we use for brewing purposes. For example, barley, rice, wheat, corn, and other cereals. Starch consists of two types of molecule. One is amylose and another is amylopectin. So amylose is the internal substance of the starch granule. It constitutes 20 to 30% of the structure by weight. It's in a lower proportion than amylopectin. Its unbranched helical chain is always with alpha 1,4 bond. So whatever is unbranched is always with alpha 1,4 bond. If it is branched, it will contain alpha 1,6 bond, like in amylopectin here. So amylopectin is the outer layer of the starch granule. It constitutes 70 to 80% of the structure by weight. So major portion of starch is amylopectin. It contains up to 6,000 glucose unit with both alpha 1,4 unbranched helical chain and alpha 1,6 branches. The branches are always in between 15 to 30 glucose units. So if you see here, amylose is a small 200 to 400 glucose unit, whereas amylopectin are larger with both helical unbranched and branched chain. Amylose are simpler molecule in a small proportion of around 20 to 30% and is only helical with unbranched chain. In the figure, if you see here, like before, this is always, if you see, this is the oxygen atom here, 1, 2, 3, 4, 5 and 6. Similarly here, 1, 2, 3, 4, 5 and 6. So the amylose is always between 1 and 4 carbon atom. So this glycosidic bond is called alpha 1,4 bonds, and amylose only has alpha 1,4 bond. Whereas in case of amylopectin, if you see here, this is the 1,4 bond, whereas this here is 1,6 bond. For example, 1, 2, 3, 4, 5, 6. The glycosidic bond here is between 1 carbon atom and 6 carbon atom. So this is alpha 1,6 bond, and these bonds, the alpha 1,6 bond, are generally between every 15 to 30 glucose units and are responsible for branching of amylopectin. Hemicellulose are non-starch polysaccharide polymer that form part of the structure of the plant cell wall. They have relatively amorphous cross-linked structure and form the matrix of cell wall being interposed within the cellulose component. They are not entirely composed of glucose residue, therefore is called heteropolymer. Other sugars are also present, for example, pentosanes or pentoses, therefore they are called heteropolymers. During malting, the beta-glucan component is preferentially degraded and in consequence, the proportion of residual pentosanes such as arabinose and xylose increases, the pentose sugar increases. Residual pentosanes that remain undegraded after the mashing may contribute to problems associated with very viscous wort. Pentosanes can bind water and form gum, and therefore the complex of pentosanes, when present at sufficient concentration, may impede wort runoff during wort filtration. This is the structure of hemicellulose. So if you see here, these are all with glucose units, but they have 1,3 bond, like you see here, 1, 2, 3, and 1, 1, 3 bond, or they have 1, 4 bond, like this here. Amino acid, peptide, and protein. So amino acid is basic building block of protein. There are 20 different amino acids in proteins, and they are composed of amine group, carboxylic group, COOH, and a side chain is specific to each amino acid. These amino acids are essential for yeast growth. The peptide are shorter chain of amino acid monomers, which are linked together by peptide bond. The shortest peptide are called dipeptide, 2 amino acid. Peptide are distinguished from the protein based on the size. Peptide has nearly 50 or fewer amino acids, whereas protein are longer chain of amino acid. Proteins form the structural and the catalytic function. The catalytic function are due to the enzymes. So enzymes are also proteins. If you see here, these small group, the small circle here, they are individual amino acids. These amino acids, for example, these amino acids, they have amino group, NH2 group, carboxylic group, COOH group, and a side chain, which are different for each different kind of amino acids. These amino acids are linked together by the peptide bond. So these bonds here are the peptide bond. The primary protein structure is the chain of amino acids that make up the protein. So this complete structure group of amino acids forms a protein. Enzymes. So enzymes are proteins that are catalytic function. They catalyze chemical reaction, facilitate and speed the reaction. They are not changed or consumed by the reaction. The molecules at the beginning of the process, called substrate, are converted into different molecules called product. So enzymes are specific. Each enzyme catalyzes just one reaction. The enzyme name reflects the reaction they facilitate. For example, if they break down amylose, it's called amylase. This is a complex structure of amylase. If they break down protein, they are called protease. If they break down glucan, they are called glucanase. So each enzyme reflects the name of the substrate that they act on. The structure of enzymes and their chemistry are very complex, like this one here. The enzymes are sensitive to biochemical environment, for example, temperature, pH, minerals, and other compounds. Lock and key principle implies that the substrate is like the key. The substrate here is like the key that fits into a lock exclusively on the active sites on the enzyme surface. Enzymes function by lowering the activation energy of the reaction that they catalyze. In order for any chemical reaction to occur, sufficient energy must be available to drive in. This is termed the activation energy. The reactant in enzyme catalyzed reaction termed substrate first bind to the enzyme. Substrate bind to particular sites on the enzyme molecule termed active site. They are made up of a unique combination of functional chemical group with particular arrangements such that one or a very limited number of reactant substrate molecules are able to bind. The enzyme substrate complex reduces the activation energy and the particular reaction can proceed. The products are released, returning the enzyme to its initial state and free to catalyze another reaction. The ability of the enzyme to bind one or limited number of substrate based upon the configuration of the active sites explains enzyme specificity. More recently, new hypothesis is developed called induced fit hypothesis, which is based on substrate specificity rather than enzyme specificity. Enzyme activity may depend on cofactor. Cofactor may be essential for activity but are not permanently bound. They are also called coenzymes. They are neither the part of the enzyme nor the substrate. They function as intermediaries by binding particular groups that are subsequently involved in enzyme catalyzed reaction. Example, calcium for alpha amylase. Various factors affect the enzyme activity, such as pH, temperature, and concentration of enzyme and substrate. Each enzyme has a specific range of pH and temperature where the enzyme functions at optimal activity and stability. Extremely high or low pH value generally result in complete loss of activity for most enzymes. Similarly, this is the optimal temperature. Higher temperature than the optimal temperature may result in denaturation of enzyme, where the enzyme is irreversibly changed in its structure and is no more able to function at all. Boiling destroys all enzyme. The enzymes are completely denatured at boiling temperature. Similarly, the substrate concentration also impacts the enzyme activity. If we have diluted substrate or diluted enzyme concentration, the activity are lower. The substrate concentration and enzyme concentration have to be optimal for maximum enzyme activity. Therefore, the grease-to-water ratio is very important to maintain for optimum substrate concentration or optimum enzyme concentration in order to achieve maximum degradation of the substrate. Enzymes in brewing. So brewer can use two types of enzymes. One is endogenous, native to barley malt. Most brewers use only endogenous enzyme. Some enzymes are not found in raw barley and are developed during the malting process, example alpha amylase. Whereas the other enzymes are exogenous from other sources. These enzymes are added externally for special situations, like in low carbohydrate light beer, beta amylase and limit dextrinase are used for complete degradation of starch. For high adjunct percentage, the inherent alpha amylase or beta amylase would not be sufficient to break down the starch in a high adjunct mass, so externally alpha and beta amylases are used to reduce the beta glucan content. If the malt is not friable, it's not well modified, then in that case the beta glucan can impede the Wort runoff, and therefore beta glucanase are used externally in the mass. In order to produce the gluten-free beer, certain specific endoproteases are used externally. So all the enzymes that we use externally are exogenous. Special care are made for German purity law or Reinheitsgebot, where the exogenous use of enzymes are not permitted. So enzymes in brewing are majorly categorized into cytolytic enzyme, proteolytic enzyme, and amylolytic enzyme. Cytolysis describes the degradation of supporting and structural cell wall substances in the coating of the starch-bearing cells of endosperm. They make the proteolytic and the amylolytic enzyme to act inside the cell wall. So for these enzymes to act, cytolytic enzyme have to first break down the cell wall. Certain cytolytic enzymes are beta-glucan solubilase, beta-glucanase. These enzymes are usually active at lower temperature, and therefore, if the malt is not friable enough during massing, brewers can maintain certain rest time at low temperature to activate the inherent glucanases, which shall break down the cell wall and reduce the viscosity of the mass for proteolytic and amylolytic enzyme to work. Proteolysis describes the degradation of the grain protein. They assist cytolytic enzyme by making starch more accessible for amylolytic enzyme. Amylolytic enzymes are responsible for breakdown of starch and sugar. We shall see these in more details. Cytolytic enzymes attack the structural protein and cell wall polysaccharide, especially beta-glucan. A wide degradation of structural substance during malting enables an easier enzymatic attack of the starch in the course of massing and consequently a better yield in the brewhouse. Undermodified cell wall or undermodified malt causes on one hand yield losses and on the other hand a transfer of large amount of high molecular beta-glucan in dissolved form. Mainly insufficiently degraded high molecular beta-glucan originating from the cell wall of the endosperm contribute to high viscosity. In the picture here you see the large starch granule and the small starch granule. So the cell wall has to be broken down for these starch granules to be accessible for amylolytic enzymes. Proteolytic enzymes. The barley protein is stored mostly in endosperm. It's combined with hemicellulose in the cell wall that encapsulate the starch. Therefore, like cell wall, the protein breakdown is also essential to access the starch. Its composition in Wort affect the fermentation and the beer quality. Nitrogen in brewing. Free amino nitrogen is the sum of individual Wort amino acids, ammonium ions, and small peptides, which is the critical source of nitrogen for brewing yeast. Low FAN results in low yeast propagation and the development of undesired fermentation byproduct such as diacetyl. However, excessive proteolysis results in too strong degradation of high molecular protein, the lack of sufficient quantities of high molecular protein, as well as the backlog of mid-molecular compound and certain amino acids such as lysine, arginine, and histidine has negative influence on foam stability. These wort and beer tend to have high color, and a surplus of certain amino acid could cause off flavor and poor flavor stability. In addition, beer with high amino acid content are very susceptible to beer spoiling microorganisms. Therefore, the proteolytic enzymes are very important in order to break down certain protein and also preserve certain protein. The proteolytic enzymes basically work at around 45 to 50 degrees centigrade. So if we want to have protein breakdown, we have to give rest at this temperature of around 45 to 50 degrees centigrade. Amylolytic enzymes are starch degrading enzymes and are also called diastase. However, for the amylolytic enzyme to act, certain preliminary condition needs to be fulfilled. For example, modification, gelatinization, and liquefaction. Modification involves the breakdown of cell wall by cytolytic and proteolytic enzymes for the starch to be accessible by the amylolytic enzyme. During gelatinization, starch takes on water, swells, stretches, and lose their organized structure. The mass become more viscous. Malt starch gelatinizes above 60 degrees centigrade, whereas adjuncts such as rice, maize, and sorghum require higher gelatinization temperature of about 75 to 80 degrees centigrade and is performed using separate cereal cooker and then returned to the main malt mass. After the gelatinization, liquefaction step takes in, where enzymes start to break down the starch and reduce the viscosity. Two enzymes degrade the starch, producing sugars. One is alpha amylase and one is beta amylase. Alpha amylase are not found in barley and are formed during the malting process. They are endoenzyme and acts in the interior of the starch granule. They produces mostly dextrins. Alpha amylase attacks anywhere, whereas beta amylase, they are exoenzyme, they attack the unbranched end, produces only maltose. Beta take bites. So alpha amylase is at 65 to 70 degrees centigrade. Generally the rest is given at 72 degrees centigrade for alpha amylase, also called saccharification rest, whereas beta amylase is active at around 60 to 65 degrees centigrade, where it acts on starch to produce the maltose. Beta amylase increases the attenuation of the beer. A rest at around 65 degrees centigrade for longer period will produce highly attenuated wort with lower limit extract, more alcohol, and light body beer. So beta amylase, if you see here beta amylase, they cannot hydrolyze alpha-1,4 linkages that are close to alpha-1,6 branch point. So if you remember, the branching is alpha-1,6 bond and the other linear chain has alpha-1,4 bond. Both alpha and beta, they act on alpha-1,4 bond, whereas alpha attack anywhere, so they go inside and attack, whereas beta bites, so they produce maltose. They always starts from the terminal end and produces maltose. Some part of starch, like this one here, near the branching point always remains, and those remaining starch are called limit dextrin. Limit dextrinase is another group of starch degrading enzyme, also known as debranching enzymes. So they act on these branched portion. Where wort with high fermentability are required, it is common to add a supplement of a preparation of microbial limit dextrinase. Endogenous limit dextrinase during mashing is highly dependent upon the condition imposed during kilning, during the malting process, and on barley variety. However, some activity of limit dextrinase persists into mashing at lower temperature of around 55 to 60 degrees centigrade. So if we want to have highly attenuated beer, the rest is given at around 55 to 60 degrees centigrade for some inherent limit dextrinase to act. To confirm the completion of starch hydrolysis, iodine test is most practiced and easiest method. Starch has a powerful color reaction with iodine solution. Amylose yields an intense deep blue to black color because the iodine incorporates itself easily into the helical structure of amylose, whereas AMYLOPECTIN gives considerably less intense blue to violet color because it lacks the extensive helical regions. Exhaustive action of beta amylase on AMYLOPECTIN leaves limit dextrin, which gives brick red or brown color with iodine. Total mass saccharification causes no change in the color of the iodine. So if you see in the initial stage, if we put a drop of iodine on the mass, it gives very dark blue to black color, whereas in the intermediate stage, the color fades, and at the end of the saccharification, iodine maintains its color and there is no change or very slight change in the color of iodine. This confirms that saccharification has been completed. And this test is one of the most used tests to confirm the completion of the starch degrading enzymes. So by this time, you should be able to answer all of these questions. I hope we were able to cover all these topics in this presentation. If you guys have any questions, you may email at brewing.distillingprofessionals@gmail.com. Hope you guys enjoyed. Hope it was an informative session. Thank you all. Cheers.